Hi, my name is Jashun Palaru from William Henry Harrison High School, and I wanted to take a few minutes today to talk about how I settled on a project, how it helped me to grow as a scientist, and what I believe the significance of my work is. The way I settled on the project I would be doing initially was a little bit unconventional, because I knew that I would be working with cancer immediately, as to me, it seemed like there were several avenues that had been, up until this point, underexplored, like, for example, the unfolded protein response, or the UPR. And the UPR is regulated by three main effectors, which are activated under cellular stress conditions and will determine whether the cell goes on to live or die under those conditions. And these pathways are overactivated in cancerous cells, which contributes to their increased resistance to stress conditions if those proteins are always active and always binding to proteins that are misfolded. And as a result, it prevents cells from dying when they should be. And these effectors are chaperoned by one protein, GRP78 or BIP, which can bind to them and inactivate them unless stress is at a suitable level for the proteins to be activated. So in my mind, I already knew that BIP was going to be one of the targets of my project, one of the main players, because it's a potential target for cancer. As if it can regulate effectors and therefore manage the UPR better, then cancer cells are going to be more susceptible. But I didn't know much about BIP and how it interacted with other molecules inside the cell. So I wanted to take the opportunity to research in a lab which focused on post-translational modifications to understand that first. Post-translational modifications are the covalent attachment of some molecule to a target protein, which will then go on to affect its shape and cause a change in its function as a result. Specifically, I focused on ampylation, which was the attachment of an adenosine monophosphate, which is pretty similar to the much better characterized modification phosphorylation, which is the attachment of a phosphate. And there are only a handful, unlike phosphorylators, there are only a handful of ampylators, perhaps 3 or 4, but they are not yet very well characterized, except for one, for example, selenoprotein O. However, the one that is characterized I found interesting because it does interact with BIP, and this protein is Huntington yeast interacting partner E, or HYPE for short. And this was relatively recently discovered, perhaps a decade or so. But there are articles as recently as June of 2021 that examine the mechanism of its function as an ampylator because it's not fully understood yet. But what we do know is on top of BIP being a natural substrate of HYPE, this ampylation is thought to have a regulatory function, which would thus place HYPE at the very top of the UPR regulatory pathway. There is one piece of the puzzle that was missing, though, from my project, and that was a definitive link between BIP and cancerous cells. And under stress conditions in cancerous cells, interestingly, BIP can be translocated to the plasma membrane of the cell, where it can act as a signaling receptor, but this will not occur in healthy, or at least non-cancerous cells. And if BIP is then preferentially expressed at the cell's surface, it cannot do its job of regulating the UPR effectively. That's an intracellular process. And thus, we have a direct connection between translocation and cancerous cell survival, and BIP and cancer cell survival, and therefore HYPE and cancer cell survival. And how exactly this occurs is not fully understood. So I sought to clarify that interaction. I hypothesized that HYPE, or specifically HYPE-mediated ampylation, had a role in that translocation. And that was what I set out to answer with this project. I asked if HYPE-mediated ampylation had a role in the translocation of BIP to the plasma membrane. So that was the motivation behind the project. Now came the actual project. Actually answering that question, asking if HYPE-mediated ampylation actually had a role, was a much more daunting task, because even though I already had some guided experience with experimental design prior to this project, this one I was going to be tackling entirely on my own. And so I started from square one, as all scientists do. I read papers obsessively for a few weeks. And I ended up finding a couple influential papers that outlined methods, the most useful of which was a paper by Dr. Amy Lee that examined translocation and outlined several basic experimental procedures which I would end up modifying for use in my own project. I had to learn several new experimental techniques, though, that weren't necessarily covered in those papers, like site-directed mutagenesis, so I could create those mutated constructs of HYPE that would then be able to test varying levels of ampylation, or culture cells so that I could examine them more closely and actually have fodder for my experiments. and lipofectamine transfection so I could cause my cells to, at least transiently, express the DNA that I had mutated and then purified. But those were just a handful of techniques on top of the many that I had. And overall, even though I learned a lot of experimental techniques and I became a much better scientist as far as experimental design, the purpose of this project was not just to benefit me as a researcher, but rather it was to fill a knowledge gap. And I believe that I successfully did that because this project, at least a little bit, serves to elucidate the function of hype in the cell, and it also lends itself easily to future experiments to begin, bridging those gaps in our knowledge, such as, for example, a mechanistic study of how exactly the ampylation of BIP affects translocation, or how activators or inhibitors of hype's natural activity, as identified by perhaps a small molecule drug screen, would affect the survival of cancer cells. So it is my hope that this work will eventually lead to breakthroughs on not just cancer treatment or neurodegenerative disorders that hype has also been implicated in, but also shine light on a protein which is incredibly important but still not fully understood. Thank you.